Okay, I think we'll make a start now. So good afternoon, everyone, and thank you for joining us on this sunny afternoon. So I'm going to start by introducing myself. So my name's Caroline. I'm a Veterinary Investigation Officer in Carmarthen Veterinary Investigation Centre. I graduated in 2004 and have been a VIO since 2018. And prior to that, I worked in practice in West Wales for 14 years. So Harriet and I have put together this session really to showcase what we do at our veterinary investigation centres and quite importantly, why we do it. You can also see our sort of approach to cases, and we want to help you to access our resources that are available to you as well, and hopefully encourage engagement. So this afternoon's session, as you can see, we're going to cover surveillance, common conditions in smallholder chickens, bovine abortion, pulpy kidney disease, and then we're going to have an interactive quiz. So a few housekeeping details before we start. This session is being recorded and recordings will be made available on our APHA YouTube channel at a later date. If you can put any questions you've got into the chat and we'll try and address them at the end of each session, time permitting, but also please feel free to contact us directly if you've got any queries after this meeting. So our first speaker is Harriet, and Harriet's going to talk about surveillance. So Harriet graduated in 2014 and worked in practice for 3 years before joining Starcroft's Veterinary Investigation Centre as a veterinary investigation officer. During her time as a VIO, she's also completed an MSc in One Health from the University of Edinburgh, and she is a member of the Cattle Expert Group. So over to you, Harriet.
Thanks very much, Caroline. Hi, everybody. I'll just turn off my camera to present. So this afternoon, I'm going to be talking to you about surveillance at APHA and the role of veterinary investigation centres. And as Caroline said, I'm based at APHA Starcross down in Devon and have been here since 2017. So first of all, what is surveillance? Well, the WHO has described surveillance as the ongoing systematic collection, analysis and interpretation of data and the dissemination of information to those who need to know in order to take action. Scanning surveillance in particular looks to detect new, unusual or changing patterns of disease, and these are what we term new and re-emerging threats. So we don't only carry out scanning surveillance to detect new and re-emerging threats. We also want to gather evidence to prove freedom from certain diseases to allow us to trade internationally, but also to generate data on some of our endemic diseases which affect the health and welfare of livestock in Great Britain. So what are we actually looking for? Well, in the middle of the diagram there are our new and re-emerging threats, but what might these consist of? Well, the top one, always the first one that comes to mind is notifiable disease. So these are diseases that vets and livestock keepers are legally obliged to report suspicion of to APHA, and we have to notify the OIE and European Commission. These can affect trade, public health, animal welfare, and also wider society, for example, rural businesses. Now, some of these are endemic, such as bovine TB, but lots of notifiable diseases are also exotic, so they're not currently seen in Great Britain. Another example of a new and re-emerging threat is a change in pattern or trend of an endemic disease, so a disease we already have, for example, Johne's in cattle. It could be a disease or infection in animals with a human health concern, for example, our zoonotic diseases and toxicities. It could be a new disease or pathogen, or a new strain of an infection already present in Great Britain. Finally, it could be a new, rare or unusual resistance to veterinary medicines. So how do we deliver scanning surveillance in England and Wales? Well, a major part of scanning surveillance is the provision of a non-statutory, subsidised diagnostic post-mortem examination service, and this makes up the scanning surveillance network. So you can see on the map that this is delivered through the Veterinary Investigation Centre network in England and Wales. And so all of the dots on the map are either where there is a VIC or a laboratory, so they're based at Lasswade and Weybridge, or our partner post-mortem providers who carry out these diagnostic post-mortems on our behalf. If the farmers live within an hour of one of these centres, then they are obliged to drop the carcass off themselves. But if they live outside this area, in the yellow areas of the map, they are offered a free-to-farmer carcass collection in those parts of England and Wales. And when a vet wants to submit a post-mortem to us, they ring their local VIC or partner post-mortem provider, and triage and risk assessment is carried out by the veterinary staff at these centres. And this allows us to select relevant cases, but also helps highlight potentially unusual disease incidents. For example, where there's high morbidity or mortality, where there's treatment failures, or perhaps an unusual presentation of disease. And all of this information can then be raised within the Surveillance Intelligence Unit, which oversees scanning surveillance at APHA. Next, we'll look at the surveillance pyramid, because this really demonstrates that everybody has a part to play. So it's important to remember that scanning surveillance provides part of the picture, but we're reliant on the work of others to bring cases to our attention. So if we start at the bottom of the pyramid, if you consider all of the animals in Great Britain, only some of these will be diseased. Of the diseased animals, only some of these will be detected by the farmer. And at this stage, the farmer may record a clinical syndrome, for example, a lame sheep, and carry out their own interventions in the first instance. However, if we go up the triangle, a proportion of those detected will then be examined by the private veterinary surgeon, or PVS. Again, the PVS may be able to make their diagnosis based on the clinical examination, or attribute a clinical syndrome such as a lame sheep. And so only some of those animals will require sampling, either live or at post-mortem examination. And of the samples and carcasses available, not all of them will be sent to an APHA lab or partner post-mortem provider. Finally, of the samples or carcasses received by APHA, not all will have a diagnosis made, which may be due to a number of different factors. So we have to be aware that what we're seeing through our scanning surveillance might not be truly representative of what's going on in Great Britain. So this is where veterinary investigation centres have a role to play. The majority of our work is post-mortem examinations of livestock and wildlife species, dealing with postal samples, for example, blood samples or swabs that vets have taken from animals on farm, but also offering advice. And you don't have to send in a submission to access our advice or raise an alert about a potential new or re-emerging animal disease. We're here to support you. And later on in the talk, I'll give you some case studies of where this has happened. If you do want to submit a carcass submission, then we discuss each of these with the private practitioner prior to submission. Carcass submissions are subsidised, so the post-mortem examination is carried out by APHA vets under a set fee, with all testing then carried out included under this charge to the vet and farmer. So you know exactly what price you're going to get charged before the submission happens. And as I said, the veterinary investigation centres also provide a key local contact point for you. So we're here to help. There is no question that's too silly and no query too small. What happens to finalised carcass or post-mortem submissions? Well, once we've sent out our diagnostic reports to the vets that have submitted these samples to us, we assign what's called a Veterinary Investigation Diagnosis Analysis Code, or otherwise known as VIDA. Now, the VIDA codes follow specific diagnostic criteria. So, for example, for cerebrocortical necrosis in cattle, we might require a histological diagnosis to make that VIDA code relevant. So it's a mark of quality, and it means that we're all following the same diagnostic criteria to come to those final summaries. These VIDA codes are then entered into a database with all of the surveillance information that we've also gathered through the history and through the submission form that accompanies the carcass or samples. And then the VIDA data is used for a variety of surveillance outputs. So not only is it really important to look at the diagnoses we make, but also it's important to look at the diagnoses not made and why we haven't made a diagnosis. It might be because an animal's been treated with antibiotics, but there might also be no apparent reason why we've not made a diagnosis. And so these might also represent a new or re-emerging disease threat. The VIDA database is compiled of all diagnostic data from APHA and the partner post-mortem providers in England and Wales, and our colleagues at SIUC in Scotland. However, at the moment, it does not include any data from private veterinary laboratories, so this is not captured in the APHA scanning surveillance. So next we'll look at some of the outputs of the VIDA data, and these might be for industry stakeholders or internal policy advisors. Some examples might be the monthly vet record surveillance reports, the surveillance focus articles in the vet record, the quarterly reports generated by the APHA species expert groups, and also the disease dashboards, which allow private practitioners to access the VIDA data, and you can use toggling functions to look at certain diseases over time or over geographical period to get a better idea of what the data is showing in Great Britain. Now, a really useful resource is the APHA Vet Gateway website. And if you just search for APHA Vet Gateway in any search engine, then hopefully you'll be able to come across this. Now, this website holds a lot of information, so you can access all of the surveillance reports that I've just talked about through this. You can also find the contact details for your local veterinary investigation centre or partner post-mortem provider. You can access our test price lists and the submission forms required to submit those samples. There's also a postcode checker, so you can be able to tell whether a farm in your practice area is eligible for free-to-farmer carcass collection or not. There's also information on the ADTS system. So ADTS, this stands for the Animal Disease Testing Service, and this is our online paperless sample submission service. It's easy to access for private vets through a portal, and you can access test details and prices. One of the advantages of ADTS is that vets themselves can track test progress, and you can receive email notifications of your results directly without having to wait for us to send those testing results out to you. It also generates free of charge postage labels for your samples. And if you want to know more information, you can access this through the APHA Vet Gateway website or just give your local VIC a ring. So I'm going to finish off this presentation by talking about some disease surveillance stories from past and present to give you an idea about what types of new and re-emerging threats we come across. So the first case here is a case of facial paralysis in calves. So a private vet gave Carmarthen VIC a ring about facial paralysis in multiple milk-fed calves on a dairy farm. The calves were between 5 and 7 weeks old, and 6 out of 15 were affected. And you can see in the picture they had a kind of unilateral paralysis of the face, affecting the ear, the eye, and the nostrils. A carcass was submitted for post-mortem examination at Carmarthen VIC, and the APHA vet found generalised lymphadenopathy and enlarged tonsils. And they were half expecting to find perhaps middle ear disease that would account for this ear drooping, but they didn't. Blue tongue virus was ruled out as a notifiable disease, and subsidised testing was carried out for bovine viral diarrhoea and malignant catarrhal fever, which was negative. Bacteriology was also unremarkable, and mycoplasma was ruled out via culture and serology. So next, the vet carried out some histopathology on the brain and the submucosa, and this was suggestive of a toxic insult. The other interesting finding was at the post-mortem examination, the APHA vet found this crumbly black material in the abomasum, which was thought to be what's called steelwork slag. Now, steelwork slag is a byproduct of the steel industry, but it can be marketed as a liming agent or soil improver. Because this was an unusual presentation, APHA carried out a farm visit to this farm to examine some of the affected calves and to gather a bit more history. They found that previously steelwork slag had been stored in a heap in the centre of the calf shed, and they found remnants of this black crumbly material in the shed and around the outside of the shed. When they compared this case to literature of previous cases of steelwork slag toxicity, the symptoms involved and the testing results matched, and so a diagnosis was made that this was steelwork slag toxicity, and we think that the kind of causative agent really is vanadium toxicity. So this allowed the APHA vet to give some advice to the farmer, and the shed was swept and rebedded, and no more cases were seen. However, steelwork slag is used as a liming agent, and the use of lime is increasing in context of increasing fertiliser costs, because farmers are looking for perhaps alternative and cheaper options to fertilise their crops. So there is the potential to see more of these steelwork slag or vanadium toxicity cases across Great Britain unless care is taken to prevent livestock exposure. And this prompted APHA to publish this case and present this case in multiple different ways to try and raise awareness in the industry of the potential toxicity of this product, and give the advice that products should be ploughed into the land or livestock should be excluded until no visible trace of steelwork slag remained on the pasture. Next, we'll look at a case involving the swine dysentery dashboard. So swine dysentery is caused by Brachyspira hyodysenteriae, and this causes severe colitis in pigs. Now, the dashboard was, this is an endemic disease, but it's also a major concern to pig health, welfare and productivity in Great Britain. So it's a really important endemic disease to monitor. The dashboard was launched in 2017, and this, as I said, contains data on the number of diagnoses made by APHA, the partner postmortem providers and SIUC, but this dashboard also included sequencing data, which can be really useful for epidemiological investigations and can give information on, for example, the origin of infection and the antimicrobial resistance of the strain. Now, this dashboard showed that between 2017 and 2019, across Great Britain, there was an increase in swine dysentery cases. And APHA found 8 known different strains of Brachyspira hyodysenteriae, suggesting it wasn't from a single source. And also, there was generally little antimicrobial sensitivity issues. So because of this uptick in cases, a industry response was stimulated. And there were a number of campaigns that were launched by industry and a number of articles put into farming press. For example, AHDB, this poster here highlights the importance of lorry washing, and communications were launched to increase awareness of swine dysentery, and testing was increased across the industry. And this resulted in the diagnostic rate reducing significantly as a result of protecting the health and welfare of pigs in Great Britain. Here's a picture of the Brachyspira dashboard that you can access as private vets if you're interested in doing so. Next is a case of antimicrobial resistance in wild seals. So we do deal with wildlife cases as well. And this case is regarding 3 juvenile British seals which are stranded on the east coast of England in 2012. Now, these were submitted to rescue centres where, unfortunately, the animals were put to sleep, and postmortems were carried out external to APHA, where the vets found abscesses in the neck, the umbilicus, peritonitis, and a pleurisy. The private vets submitted swabs from all 3 cases to APHA for bacteriology testing, where we isolated Klebsiella pneumoniae. Now, this bacterial strain showed multiple antibiotic resistance in all 3 cases, and the resistance patterns and subsequent strain typing suggested a human origin for infection. And then consulting literature and known strains, it was suspected that this had originated from sewage outflows into the coastal environment. So this is an interesting case because this basically added to the evidence base demonstrating the role of wildlife as sentinels of health risks to humans around water. And again, this was published and awareness was raised regarding this issue. And this is important to highlight because the postmortems here were not done by APHA, but we were still able to access this surveillance data and act on this new and re-emerging threat. And finally, we'll talk about ulcerative skin lesions in sheep. So these were first reported by APHA in 2008 and then again in 2019. But this is truly an emerging condition. So usually these sheep present with ulcerative skin lesions and cellulitis, often affecting the lower limbs. However, they're not always lame. They appear to be contagious with how they spread through flocks, and they resemble strawberry foot rot. However, parapox virus and dermatophilus, which are often implicated as the causes of strawberry foot rot, have not been detected in these clinical cases. They seem to respond to antibiotics, so there is potentially a bacterial component. However, we don't know what the cause of these is. And these are, again, a good demonstration of how APHA can get involved in cases, because these sheep, A, often don't present as lame, and B, often respond very well to antibiotics. So in many cases, there is not a carcass to post-mortem, and there might not even be swabs to culture. But if you have any of these cases or anything else that you think is interesting, then it's really important to give your local VIC a ring about these, and we can help you investigate them further. So finally, I'd just like to acknowledge my colleagues Ed and Liz, who helped me put these slides together, and the multiple vets and species expert group leads at APHA for their input into these cases. Thank you so much for listening, and if you have any questions, I'll be happy to answer these.